Okay, so welcome back. We're in lecture 2 here. So this is going to be on mutations, and this is, I find very, very interesting. Now, the big thing here is mutations can happen a number of different ways. Okay, and what we talked about in grade 11 primarily was chromosomal mutations. We did talk a little bit about point mutations, but chromosomal mutations. Remember, I'll kind of just draw something back here. We'll see if we can see it here. So here's a chromosome. Okay, and on a chromosome, you've got hundreds of genes. Hundreds of genes. So if you have an error in a chromosome, you're going to have lots of problems, okay? Because you're going to have, if you have, say, an extra chromosome, you have an extra of all of these genes. Now, an extra chromosome would really be 3 because we normally have 2. But this would be considered a trisomy disorder, okay? Where really you're only supposed to have a little bit of protein, okay? And because you have 3 chromosomes, you're going to have a ton of protein. An example of this would be like someone who has Down syndrome. Okay? And someone who has Down syndrome has multiple different difficulties, multiple different abnormal protein levels, which is causing a number of disorders in people who have Down syndrome. As opposed to, say, someone who has cystic fibrosis, which has very limited number of proteins that are malfunctioning compared to someone who has extra chromosomes. So usually when you have extra chromosomes or you're missing chromosomes or you have an error in an entire chromosome, you're usually born as a stillborn, okay, or as a miscarriage. It's very, very difficult to have abnormal protein levels, and to have an entire chromosome of abnormal protein levels makes it very difficult. That's why people with Down syndrome are so remarkable. They're so remarkable because they're very, very functioning. They can do just unbelievable things, yet they still have this pretty severe error. Okay, so let's go through mutations today. So I'm just going to take a look here, and here we go. Okay, so here's mutations. So here's the chromosome I was talking about. Okay, so this looks like a chromosome 9, and one of the errors you could have is you could have this whole chromosome duplicate, okay, or you could be missing one. So an extra one would be a trisomy from grade 11, and if you're missing one, it'd be a monosomy. Okay, so what is a mutation? A mutation is an error in the DNA sequence that happens during replication. Okay? So most of the time, natural errors or natural mutations are during replication. We can have other errors that are induced, and we'll talk about that a little bit later. Now, since the error is now part of your DNA, all cells that are created from this cell will contain the error. And that's the big problem. I mean, it's a big problem, especially in cancer. Because once one of your cells gets cancer and it's got an error, okay, every cell that's made from that cell is going to contain that error because it's right in your DNA. Okay, so your instruction booklet, your main booklet now has an error in it, and that is major. It's not like an mRNA that has an error, because that's not a big deal. But if your DNA, your original book that has all of your information, gets an error, then it's a big deal. Okay, now, just a couple things. If replication error occurs in the testes or ovaries, the error will be in the gametes and can be passed on to their offspring. Okay, so that would be like passing on a disorder, like passing on cystic fibrosis, passing on Huntington's disorder. Okay, those would be errors that you can pass on to your offspring. Now, passing on something like cancer is a little bit different. Okay, that'd be very difficult to pass on. You could pass on the predisposition to develop cancer, but you most likely wouldn't be passing on the disease cancer because most people develop cancer after they're already born. And unless those cells that have the cancer have made it to your sex cells, most likely your sperm and your eggs will be cancer-free. Okay. Now, if the error is in the somatic cell, the error will die with the individual. So if it's kept out of there like a cancer cell and you don't have cancer cells inside your sperm or inside your eggs, then that cancer cell, that mutation, is going to die with you. Okay, so are all mutations harmful? No. Okay. Some of them are harmful. Okay? Some of them are very harmful, leads to the development of diseases like cystic fibrosis. Okay? So most cases, if you have a mutation, it's either going to be harmful, it's going to hurt you, or it's going to be no effect. It'll be undetected. Okay? And so a mutated gene may be masked by the normal gene carried on the second chromosome. If you remember, someone like a carrier of cystic fibrosis, you remember dominant and recessive from grade 11. So most of the time, it's either harmful if you have a mutation or undetected. Now, there is rare times, and this is what's caused evolution, that you have a positive mutation. And if we didn't have mutations, we'd still be bacteria, okay? And there'd still be only bacteria on Earth. But the fact that we've mutated, and some of these mutations have been positive, it's allowed us to evolve, and that's the whole idea behind evolution and what we've developed into. Okay, so we're going to categorize mutations into two, and we're really going to focus here on point mutations. But we also have chromosomal mutations, and we covered these a lot in grade 11, so I'm really going to kind of focus on the point mutations. And a point mutation means that, you know, a gene has been has an error in it, where a chromosome means the entire chromosome or portions of it have the errors. Okay, so within point mutations, and these are, you know, pointations or a mutation on a specific gene, we've got three: substitutions, insertions, and deletions. Now, substitutions are not going to be half as bad as these insertions and deletions. Okay, substitutions are not horrible. I mean, they're not good. None of these are good, but substitutions you'd way prefer over an insertion and deletion. I'll show you why. So what a substitution is simply you sub out one letter for another. Okay, so say originally it was AAT, now I've got AGT. Okay, but notice everything else downstream is everything perfectly fine. Okay, this is all lined up perfectly. Everything up here is lined up perfectly because all I've done is subbed out one letter for another letter. Okay, now when you do this, okay, there could be three results. So if I sub out a letter, I could have a missense, which means that it's going to alter one of the amino acids. Okay, so if my original thing was the fat cat ate the wee rat, and this is an amino acid, amino acid, amino acid, amino acid, amino acid, amino acid. These are each codons. Okay, the substitution had an effect at one amino acid. So this amino acid is fine. This one is. 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 But one amino acid is off. So an example of that is sickle cell anemia. So here's the original DNA code. Okay, and here's the original amino acid sequence. Well, if we have a substitution in just one base pair, okay, and this base pair is prone to substitution, which is why, you know, sickle cell anemia happens to more than one person. It's a disease because this one's prone to having this error. But if I sub out these AT and it switches them, so it goes, you subbed out this T for an A, and that caused this one to become a T. Notice that my one amino acid is different. It was glutamic, now it's valine. Right? Every other amino acid is perfectly fine, but this one is off. Right? And that causes red blood cells to turn from our normal circular shape that can carry lots of oxygen into the sickle cell shape, which carries low levels of oxygen. Right? So this is kind of a little bit more of a serious effect, even though it's substitution. So that's one effect from a substitution, is a missense. The other one, and this is very bad, is a nonsense. Okay? A nonsense puts a premature stop codon in there. So it basically alters the DNA sequence. So here's our DNA sequence, and it changes, one of the substitutions accidentally puts in a premature stop codon. So what happens there is the amino acid produces, or basically there's not enough amino acids. It produces a short non-functional protein. So instead of making the full protein with all these amino acids here, each one representing amino acid, each word, you shorten it up. Okay? Normally it's lethal. The protein's going to be non-functional. So this is kind of a horrible substitution. Okay? So last one is the best one, silent mutation. You won't even know what happened to you. It has no effect on the final protein. Okay? It means that the error could have occurred on the intron region or the non-coding region, or in the third base of the codon. Okay? And that third base is kind of considered what we call a wobble position, because you can swap out the third base in a lot of cases and still make the same amino acid. So if I had CAU, and then I had a substitution, and the UNC and the UNC switched, I'd still make histine, and I'd still have a fine protein. Okay, so that's the best one. All right, so here's our different types. A silent mutation, you know, the effect had no change on it. Okay, where, you know, we changed an A instead of a G, but it still made my glycine, so everything was perfect. Okay, here's a missense, which, you know, an amino acid's been swapped out. It was glycine, now it's serine, but everything else is perfectly fine. Here is the worst, is when you have a premature stop codon. It's definitely going to be non-functional. Okay, so we've gone through substitutions. Now we're going through the brutal ones, insertions and deletions. Okay, these are almost always detrimental. So when we had an insertion, an extra base is placed into the DNA, and this changes the reading frame, aka it causes a frame shift. Now a frame shift is going to be caused by an insertion or a deletion, okay? And both things are going to cause some major problems. It causes a different amino acid to be translated after the insertion. So here we inserted a letter here. So this amino acid right here is fine, this amino acid is fine, this amino acid. But starting here, every amino acid downstream is going to be screwed up, right? Because everything is now shifted downwards. So it should be ATE, but because this extra letter is now in here, this is the new codon. So these 3 letters are going to make an amino acid, and then these 3 letters are going to make an amino acid, and then these 3 letters are going to make an amino acid. So it really screws up the amino acid sequence, okay? Almost as bad as putting a premature stop codon. Okay, depending on the severity of the change, but it's just almost as bad. A deletion will also cause a frame shift. Okay, so if I delete a letter, so here I deleted the C, and if I delete the C, everything moves down, and now everything past this point is all going to have a screwed up amino acid sequence. Okay, the first 2 are fine, but everything past the deletion is screwed up. So really, with the frame shift, it's very important to know where that frame shift happened. And a frame shift is another word for insertion and deletion. Because if it happened very late, it's not as serious. If it happens very early, it's very serious. All right. So here's an example of a frame shift. So here's our normal sequence. I put in an insertion. I added an extra U. That's going to change this amino acid from tyrosine to leucine, because instead of UAC, because I have that extra U, it's going to be UUA. And then that's going to change this one from glycine to arginine, and then so on all the way down. Okay? Deletion, same kind of thing. Wherever the deletion happens, it's going to screw everything up from downstream. So in this case, it actually made a premature stop codon, okay? Which also can happen as well, which is going to be just as bad. So here's the idea. So here's our normal strand. This is what we want to produce: methane, lysine, PHE, and glycine. Okay. As soon as we have a missense mutation, so a missense means that you're going to swap out one of the amino acids. Okay. This is going to be a premature stop codon, which we call a nonsense. And then if you insert a letter or delete a letter, it's called a frame shift. And those are our 4 main types of mutations. Okay. So point mutations, although the results can vary, generally speaking, in terms of severity, the least severe is obviously going to be a silent. Missense, where you make the wrong amino acid sequence, okay, is usually not as bad because your protein is still functional. It just has one amino acid swapped out. Now, it can be really bad in certain cases, depending on what that protein does for your body. If it's a really important protein, it can be very serious. If it's not a very important protein, it can be less serious. Okay? Nonsense, okay, and that's when you're going to have a premature stop codon. And again, it depends on its severity. Like it might be where you don't have hair color anymore, where you have white hair. Well, that's a mutation, that you're not producing the protein to make your hair dark anymore. And I mean, it's not lethal, so that's why it sometimes depends on the importance of the protein. Okay? And then frame shift is usually thought of as the most detrimental. I mean, nonsense could be just as detrimental as a frame shift. It really, between these two, are definitely going to be the top 2, and these 2 are going to be the bottom 2. Okay. Now, the last one is chromosomal mutations. I'm not going to talk too much about these. I'm not going to ask you a lot about them. The big thing with chromosomal mutations is you're going to have segments of DNA. You're going to have segments of genes. Several genes are going to be, different. You're going to have lots of different protein levels than what you should. Okay? So the first mutation that can happen in chromosomes is a translocation, and that's simply part of a chromosome coming off of, say, chromosome 4 and then attaching to chromosome 20. So it's like where one chromosome leaves and then goes and attaches to another chromosome. Now that's a problem because when DNA goes to try and make the protein, it goes to look for chromosome 4 and says, oh, there's no genes here. And it doesn't know where the genes are. It doesn't know that it went over to 20, so it basically doesn't make the protein. An inversion, okay, an inversion is basically just a piece of DNA flipping. So it would be like this piece of chromosome coming off, inverting, and then reattaching to chromosome 4. So it's just upside down. It's usually not as bad. It depends on where the break occurs. Okay? and I'm not going to ask you anything about those. Okay, last thing I do want to talk about is causes of mutation, though. So what causes mutation? They can be spontaneous. Okay, and that means DNA polymerase 1 or 3 missed a mistake during proofreading. So that's just during normal replication, you spontaneously have a mutation. If you have a freckle or a mole, you have a spontaneous mutation. So you look at your freckle and you say, one of my skin cells, when it replicated, mutated. And that's what my freckle is. It's a mutated skin cell or a mole. Okay? And if that thing's not replicating anymore, it's not a big deal. But if you find your mole getting bigger and bigger and bigger, okay, and it's irregular shaped, you should probably talk to your doctor about that and have him or her look at it. And they'll probably monitor your mole to make sure that that air is not spreading throughout your skin. Okay, the other way that we can get a mutation is through induced. Okay, and it's an error exposed by exposure to a mutagenic agent like UV radiation or X-rays, or pesticides or cigarette smoke. Okay, things that induce errors are called mutagens. So we're going to go through some. So we've got physical, biological, and chemical. The first one is physical. Okay? And they are considered things like X-rays and UV damage. Okay? And they're mutations caused by the physical tearing of DNA. So X-rays come in and start basically tearing your DNA apart. Okay? It can cause all kinds of point, frame shift, and chromosomal errors. Okay? X-rays, so that's why you can't get a lot of X-rays. You get one X-rays, and they're not going to be able to give you, another X-ray, another X-ray. You can get a limited amount. Same with like CT scans. Okay? You can't get a ton of them because they don't want to expose you to all this radiation. Okay. And then same with the sun. Okay, chronic exposure to the sun, UV comes in, okay, goes inside your skin and basically causes a reaction mostly between C and T, and can cause melanoma. The other things we have, if it's not going to be physical, it could be chemical, and these are mutations caused by chemicals that enter the nucleus and cause the cell disruptions. They can cause substitutions or frame shifts. The most common are nitrates, gasoline fumes, pure carbon. So like when you eat like a burnt hot dog, okay, or burnt toast or a burnt steak, all of that burnt stuff is pure carbon. Okay? And when you eat that, it is a carcinogen. Okay? And then there's a ton in cigarettes. So if any of you out there are smoking, you're crazy, okay? Because you're just going to be mutating your DNA, and why would you want that? Okay? And then these are carcinogens. These are other chemical things. I mean, when people say everything causes cancer, there's some truth to that. There's a lot of things that can cause cancer. And the last one I'm going to talk about is biological. So these are viruses and bacteria can also cause mutations in your DNA. They most commonly cause substitutions and frame shifts. The most common is HPV. Okay, and any of you females out there, you've probably been vaccinated against it because it is a virus that can cause cervical as well as ovarian cancer. So we get this vaccination, or females get the vaccination for HPV, so you don't get the virus, and then you have a decreased chance of developing ovarian cancer. Okay? Another bacteria that can cause cancer is called H. pylori. It gets into your stomach. Okay? It often causes GI disorders, specifically ulcers, and can also cause GI tract cancers. Okay, so repairing DNA, I'm going to ask you to go over this on your own. Okay, we have two different types of repairing DNA. We got photo repair for UV, and then we have a non-specific, but we have that covered in the content. And that's it. There's a couple links here that you can take a look at if you like, and then you don't have to do that project. That's from another for another class. But I hope this helped clarify things. Make sure you email me with any questions or concerns. Okay, we only have one more topic to go through, and then we're going to begin reviewing for our test Tuesday.